If you're a Year 13 A-level biology student and you've been learning about aerobic respiration, then this video is for you. I'm going to focus here on the Krebs cycle. I have already done glycolysis and I've already done the Lynx reaction, and I will do oxidative phosphorylation, so please do check out those other videos. But this one, we're going to focus on that Krebs cycle and what happens when we get to this stage. So just as a quick recap though, glycolysis produced pyruvate. Link reaction changed the pyruvate into the acetyl coenzyme A. And now we are going to go on to the Krebs cycle. So it's important to understand that the Krebs cycle happens inside the mitochondrial matrix. Now, the acetyl coenzyme A, the 2-carbon acetate that has been picked up by the coenzyme A, is going to be delivered to the Krebs cycle. And what's going to happen is our 4-carbon molecule, oxaloacetate, is going to fix that 2-carbon acetyl group to it, and we're going to result in a 6-carbon molecule called citrate. So the coenzyme A, as I like to call it, is a taxi. It's picked up the acetyl group from the Lynx reaction and it has delivered it to the Krebs cycle, and then the coenzyme A can go back to the Link reaction to do it all over again. So the oxaloacetate, the 4-carbon molecule, has now had a 2-carbon molecule added to it, and that has given us our new molecule, which is our citrate. And what needs to happen in our Krebs cycle is that citrate molecule is first of all going to be decarboxylated. Now, DE, removal of the carbon dioxide, therefore decarboxylated. We're going to take the 6-carbon molecule down to a 5-carbon molecule. Now, we're also going to oxidate the molecule, and to do that, we need to reduce another coenzyme. So NAD plus comes in and it becomes reduced NAD, and it has picked up the hydrogens, which has oxidized that molecule, and we now have this new 5-carbon molecule. Now, you do not need to know the intermediate stages if you're studying OCRA or if you're studying AQA. You do not need to know those intermediate stages. You just need to know the 6-carbon molecule citrate decarboxylated into a 5-carbon molecule, and then that 5-carbon molecule is going to be decarboxylated again into a 4-carbon molecule. So what we've got again is this 5-carbon molecule, as it's decarboxylated and becoming the 4-carbon molecule, again it is going to be oxidized. For it to be oxidized, the coenzyme NAD plus has to be reduced. So we've now got a 4-carbon molecule, and what's going to happen is we're going to have a bit of substrate level phosphorylation occurring, where that 4-carbon molecule is going to lose a phosphate, and that phosphate is going to be added onto ATP. It's going to be transferred. Therefore, the intermediate molecule is transferring the phosphate to the ATP molecule, substrate level phosphorylation. The 4-carbon molecule intermediates again are going to go through a few stages of oxidation. This time we've got a new coenzyme coming in. This coenzyme is FAD. So FAD comes in, it is going to remove the hydrogens, therefore becoming reduced, and that is FADH2. Another NAD is going to come in, and that NAD plus is going to be reduced to NADH. Again, we have removed 2 hydrogens. So we have taken our 6-carbon molecule, we've decarboxylated it twice, and we've also oxidized it a number of times. Now remember, for every glucose molecule, you get 2 pyruvate. For every 2 pyruvate, you get 2 acetyl groups. So therefore, the Krebs cycle happens twice. So it's important, as I always say, for us to look at our inputs and our outputs. So if we look at our inputs and our outputs then for the Krebs cycle. So input for the Krebs cycle, we have 2 acetyl coenzyme A's and the output is 2 coenzyme A's, and they can return back to the Lynx reaction to do it again, to pick up more acetyl group to take into the Kreb cycle. We've had decarboxylation happen. For every 1 acetyl group coming in, we get 2 carbon dioxides. So therefore, because we have 2 coming in for the Kreb cycle with 1 glucose molecule, there has to be 4 carbon dioxide coming out. Now, remember the Kreb cycle happens twice. There are 3 NAD pluses coming in. Now that's times 2, so that gives you 6 in total. So we have 6 reduced NADs leaving the Kreb cycle. We have 2 FADs coming in, therefore 2 FADH2s coming out, reduced FADs. And we have 2 ADPs coming in and 2 ATPs coming out. Now, it's important to remember that the Kreb cycle happens inside the mitochondrial matrix. So therefore, to go from one stage to the next, to the next, to the next, there are lots of enzymes involved and lots of intermediate parts. But as I said before, the only ones you need to be able to name is oxaloacetate, acetyl coenzyme A, and the citrate, which is the 6th carbon molecule. All the other intermediates, if they want you to know them, they're going to give it to you, but they'll expect you to apply your knowledge to that concept. So the key thing about the Kreb cycle was that the oxaloacetate was converted into citrate, and then the citrate is converted back into the oxaloacetate, and that's why it's called the Kreb cycle. Now, it's really important to note here that carbon dioxide is a waste product. So carbon dioxide is not necessary for any part of the process. So carbon dioxide will diffuse out of the mitochondrion, out of the cells, and therefore be transported in the blood back to the lungs so that we can breathe that out when we do our ventilation. The reduced NAD, the reduced FAD, is very important because they are needed for the next stage in aerobic respiration, oxidative phosphorylation. Remember, they are coenzymes. Coenzymes like taxis are picking up the hydrogens and electrons, and they're going to deliver them to the cristae where they are necessary for this next stage. So do check out my video that I will post on oxidative phosphorylation.